My name is Carolyn Coyle, and this is my presentation on the Shigella flexneri bacteria in the human small intestine. Shigella flexneri is a rod-shaped gram-negative bacteria. It is typically about 2 micrometers in length, and it is a non-mobile organism, so it relies on help from its host to spread through cells. It is one of 4 types of Shigella bacteria, and it causes the disease shigellosis in humans and other primates. Shigellosis is characterized by intestinal inflammation that leads to bloody stool or dysentery. Its human hosts experience severely painful abdominal cramping and diarrhea. Shigella flexneri enters the host's body through contaminated food and water via the fecal-oral route. Because of this means of entry, shigellosis mainly affects humans in impoverished communities where cleanliness is more difficult to maintain. This image shows the epithelium in the small intestine of the human digestive tract. The orange shapes are representative of columnar epithelial cells, which are about 25 by 8 micrometers in size, compared to Shigella flexneri's 2 micrometer size. These cells have occasional M cells in their lineup, which is shown in green in this image, and next to the M cell is a microphage shown in blue. The individual bacteria are shown in purple, and their route through the intestinal lumen to the basolateral pocket and into the epithelial cells is marked by pink arrows. The pathogenesis of Shigella flexneri bacteria begins with the bacterial cells floating through the host's intestinal lumen, where they are picked up by M cells along the epithelial surface of the intestines. Epithelial M cells usually work to improve human immunity to pathogens. They draw in some of the contents inside the intestines and create possible antibodies for those materials. Shigella flexneri, among other bacteria, use the M cell pathway to their advantage by using it to cross the follicular-associated epithelium through its junctional complexes. These M cells sit in epithelial junctions and allow passage of the bacteria from the intestinal lumen into macrophages that spit out the cell into the basolateral pocket of the epithelial cells. Here, bacteria gain access to other cells in the digestive system. From the basolateral pocket, Shigella flexneri bacteria attach to the basolateral surface on neighboring epithelial cells and prepare to secrete their virulent matter and spread through the epithelium. Shigella flexneri uses a needle-like appendage to perforate the surface of the epithelial cell and inject its infectious material into that cell in a type 3 secretion system. Since Shigella flexneri are immobile organisms, they then use the host's actin proteins to aid in movement to other cells. Actin is a contractile protein that aids in muscle contractions and other digestive contractions in the intestines. Only about 10 to 100 Shigella flexneri cells are needed to cause illness in human hosts, leading this disease to be endemic in many developing countries where clean water and antibiotics are harder to come by. The inflammation in the intestinal lining can lead to ulcers and severe pain in hosts. This is how Shigella flexneri bacteria rapidly spreads throughout the intestinal epithelium and causes widespread inflammation and dysentery in its hosts.